It's my pleasure to introduce our speaker of this afternoon. Hers is the face that you've seen around the lab since last August. And Colleen's got an office down the hall at the end of the faculty wing, and I urge you to get to know her. Colleen did most of her biological growing up in Spokane, Washington, and then most of her academic growing up at the University of Washington in Seattle. With a Bachelor's of Science in Biological Oceanography from University of Washington, magna cum laude, 2004. A Master's in Oceanography from the University of Washington in 2008, looking at chitin in cell walls of diatoms. Now this is important because I'm going to get back to a connection there. Her PhD was completed in 2012 on environmentally induced changes to the diatom cell wall and the implications of these changes on biological cycles. She then did a postdoc at WHOI with Ken Bissler, aka Mr. Thorium, if you know Ken. And then there's a connection to Moss Landing Marine Laboratories that I don't know if you have made yet, but I'm about to make it for you. Both the master's and the PhD were advised by Ginger Armbrust, and she is a Moss Landing Marine Labs alumni from the mid-'70s, now director of the School of Oceanography at University of Washington. Fancy pants position for a Moss Landing graduate, and she is doing awesome there. Just got a $35 million award to go play in biological oceanography. Ginger, as she was known, used to hang out with the Flux crowd in the Vertex program. That was Madeline Urer, my wife Susan, Bernadette Allen, Kim Pepard, and others. Later, Susan went on to do her master's at Moss Landing Marine Labs doing research in chitin flux from sediment traps. And it's an analytically challenging problem. Bissler got into thorium using the method that I developed at UC Santa Cruz. You see how the connection's going here? In addition to a purely academic interests and her pursuits, Colleen has taken an interest in public outreach and has created several videos on YouTube that you should check out. She is one of only a few brave scientists to tastefully animate defecation by copepods. At WHOI, Colleen teamed up with Ken Bissler to help organize and conduct a citizen scientist program to monitor the spread of cesium-134 and 137 from the Fukushima Daiichi nuclear disaster. And our research group has taken samples for this program on our recent cruises. So she feels like a lost sister to me. And today, you're going to hear more about her straight from the source, as Colleen tells us, about linking phytoplankton physiology and with carbon export out of the surface ocean. Please welcome Dr. Colleen Durkin.
Thank you. I did make the chitin connection to Moss Landing. All right. Well, so I'm going to tell you about research that I've been working on most recently. And the goal of all of it is to link phytoplankton with the sinking carbon flux out of the surface ocean. And the characters of this talk are shown here in this slide. First, we have the phytoplankton. So you can see there's a lot of there's diverse types of phytoplankton growing in the surface. And then we have all the particles that are sinking out of the surface ocean, which is everything on this slide. So that includes detrital particles like these organic aggregates and fecal pellets. So the way that all these things are united is by this process called the biological pump. And in case you don't all understand what the biological pump is, I'm going to show you one of these videos. So don't worry, you don't have to go to YouTube. Just so we're all on the same page.
So I hope that by seeing this you can recognize that there are some really complex processes involved in the biological pump. There's a lot of diversity in the organisms that are growing in the surface, and then there's a lot of ecological interactions and then also physical processes. So it's pretty complicated. And so the way what I'm going to talk to you about first of all is the surface ocean. So part one of this talk is I'm asking the question, how have phytoplankton adapted to thrive in changing ocean conditions? And specifically, I'm going to talk about the evolution of silicon uptake in diatoms. And then the second part of this talk is thinking about those sinking particles below the surface ocean. And the question is, what particles are actually contributing to carbon flux out of the surface ocean? And then finally, I'm just going to really briefly talk to you about what I'm working on in the future, which the goal of that is really to combine these two parts together and think about production in the surface and sinking out of the surface at the same time, so we can get a more integrated idea of the things that are affecting carbon export. So first of all, let's talk about production in the surface ocean. And what I'm going to tell you about are diatoms. So what you're looking at here is a collage of micrographs of diverse diatom species. And the reason that I'm focusing on diatoms specifically is because they are a major part of the carbon cycle. In the oceans, they're responsible for about 40% of the marine primary production. But then when you look at the global scale, including land, diatoms by themselves are responsible for 20% of global primary production. So that means they're the equivalent of the tropical rainforests. You could also flip it the other way and think about how much oxygen they're putting into the atmosphere. And that means that 1 out of every 5 breaths that you take comes from a diatom. And to me, this is just amazing because these are microscopic organisms, but they have this massive effect on global cycles. Diatoms are also really interesting because they form a silica cell wall. So this gives them a unique biogeochemical role in the oceans. And to top it all off, if that isn't enough, diatoms are also the most diverse phytoplankton group. The first diatom was, the first observation of a diatom was published in 1703. So people have been observing diatoms for more than 300 years. And there's more than 100,000 described species. So these are an extremely diverse group of phytoplankton. So how did all of that diversity evolve? This is a schematic of how diatoms evolved. The earliest diatoms may have emerged about 250 million years ago. And that's based on molecular data, so there's not actually fossil evidence, but perhaps they're that old. But the earliest diatoms that we recognize today are the centric diatoms, and they emerged about 183 million years ago. So they are the most basal type of diatom group in the modern oceans. And then the multipolar centric diatoms emerged, and I wanted to highlight that within the multipolar centric diatom group are the Thalassiocyreales, and they're just a diverged and very diverse group of diatoms within multipolar centric lineage. And the most young type of diatom are the pennates. They were the last to emerge. And the characteristic that all of these lineages have is they all precipitate a silica cell wall that's called a frustule. And so you can see here that those frustules are also just as diverse as the diatoms. These are cells that have been labeled with a fluorescent stain that incorporates into newly deposited silica.
And the different colors are really just to make them look prettier. So you can see that forming a cell wall is really an essential part of their biology. The cells can't continue to grow and divide unless they form a new cell-to-cell wall. So this process is really tightly linked with production in the surface ocean and the carbon cycle. So in order to make that cell wall, they have to take up silicon from seawater in order to grow. So how do diatoms take up silicon from seawater? Well, if we were able to look inside a diatom, so here's just an illustration of a Thalassiosaurus cell. If we could take off the top of its frustule and look inside, you see a cell membrane. So the cell membranes are located inside the frustule. And then embedded in the membrane are the silicic acid transporters, which for short are called CITs. So I'm going to be talking about CITs a lot. And that is this protein that's embedded in the cell membrane. And these proteins actively take up silicic acid from seawater. And the way that cells actually make these proteins is that they have genes embedded in their DNA. And so that's this little orange section is supposed to represent the section of the DNA that encodes a CIT gene. When they want to actually make a CIT gene, they transcribe that DNA into messenger RNA. And this is what's referred to as gene expression. So when I talk about gene expression, I'm talking about how much messenger RNA is the cell creating. And that's a signal of, that the cell wants to make more of this protein. And so then to make the protein, they translate the messenger RNA into the protein, and then the protein gets folded up and put inside of the cell membrane. So these CIT proteins are a really fundamental part of diatom biology. They all need to take up silicic acid from seawater. And we also know that diatoms are extremely diverse. So for me, the question is, how did these CIT genes evolve? How did the silicon transporters evolve in diatoms? And this is the question I'm going to answer for you today. And I want to acknowledge my collaborators on this project, Julie Kester, Sarah Bender, and Ginger Armbrust. And the main way that we went about trying to figure out how CITs evolved was we relied heavily on this relatively new genetic data set called the Marine Microbial Eukaryotic Transcriptome Sequencing Project. And this was a pretty massive sequencing effort by a lot of different groups all over the world. And after a few years of work, 650 transcriptomes from diverse marine protists were produced. And a transcriptome is all of the expressed genes in an organism. So it's similar to a genome, except that it's just the expressed genes. And this is actually a really special data set because this type of information about marine protists just didn't exist before. So we couldn't even ask these types of questions as powerfully. And I should mention that I downloaded this on the Moss Landing Marine Lab computer. So if anybody wants to look at this data set, I would love to help you do that. And then I'm also going to mention some of the bioinformatics tools that I've used, just for you people who are doing molecular research, so you know what types of tools I use. But for the rest of you, I don't want you to get too distracted by that. So how we found the CIT genes in this data set was that we identified the homologs using HMM search. So we identified CIT gene homologs. And in total, we identified 423 CIT sequences. So that included sequences in this new data set and also from previously published research. And so of those 423, 70% were newly identified. And those 70% of the new sequences represented a much, much more diverse community of marine organisms. So to identify how those sequences evolved, we made a phylogenetic tree to look for the evolutionary relationship. So I just want to show you what this tree looks like so you can appreciate how much data is there. There's no way for you to look at this and analyze what it means. But the reason I'm showing it to you is because I want you to appreciate that in the next few slides, the results I show you are based on a lot of data. And I didn't actually manually go through this tree and analyze it branch by branch either. I used Python programs to pull out the interesting patterns. So let me show you this tree in a more simplified form. What I did was I collapsed the branches so that into groups of sequences that had a common ancestor. So for example, this triangle here represents 126 sequences that all share a common ancestor. And if you're not used to looking at a phylogenetic tree, the horizontal lines are the branches, and the length of the branch indicates genetic difference. So the longer the branch, the more genetically different it is. And the vertical lines are there to connect the most closely related branches. So it's very similar to a family tree. And so, for example, this group of sequences is connected to that branch, and they have a common ancestor right there. So by grouping the sequences this way, we can identify clades. And a clade is just a group of sequences that all share a common ancestor. I'm going to start out by talking about this clade at the bottom, the white clade. And this was defined as the outgroup, and it was used to root the tree. And the reason I chose this group was because every sequence in this clade is encoded by a non-diatom organism. So all of these sequences are encoded by diatoms. So then if we look at the diatom sequences, the most basal part of the tree is this B clade, although you might notice that it's not a true clade. There's not just one common ancestor, but it is a whole bunch of sequences that all clustered at the most basal part of the tree. So I'm labeling it B. And then we have A, C, D, and E. So what I hope you appreciate is that the diatom sit sequences are extremely diverse and have really diverged. And what I would like to know is whether this is just random diversification or if it actually has some functional consequences in diatoms. So one way to do that is to model the protein functions. And so for example, we can look at the protein domains. Within this clade C, the proteins look like this, and each symbol there represents a different region of the protein that's predicted to function in a different way. So for example, these blue triangles represent regions of the protein that will probably go back and forth through the cell membrane. So you can see that this protein goes across the cell membrane quite a few times, which that's great because we know that these proteins are embedded in the cell membrane. So we can see that a very similar protein function is predicted to occur in clade C, E, and the non-diatoms, whereas in clade A and D, we get these extra functional domains on the end. And then in clade B, we get some other functional domains. And also, what was pretty exciting is there's also these sequences that are basically three sit proteins just concatenated onto each other. So there's a domain duplication. And I highlighted where those are on the tree in the gray triangles there. So how are these functional differences distributed among diatoms? Do all diatoms have all of these types of sits? Do they have all the functionality, or is it distributed differently among diatoms? So the way I'm going to show that to you is going back to this diagram of diatom evolution. And where we find those most basal sit genes is in every diatom lineage. We found it in pennates, multipolar centrics, and radial centrics, and even in this diverged Thalassiocyrales lineage. So what we think is that these sit genes were present in the earliest diatoms. They were also the most basal part of the tree, so it makes sense. Then if we look at the distribution of the clade C and D sit genes, they're also pretty widely distributed, except not in the Thalassiocyrales. So we think that they were also in an early diatom, but either it was lost or somehow didn't make it into the Thalassiocyrales lineage. And what's really interesting now is if we look at these other clades. Clade A is only found in the pennate diatoms, so it seems like this clade emerged within the pennate diatoms uniquely. And the same thing for clade E, it's only in the Thalassiocyrales. So this is pretty exciting because there are these lineage-specific sit genes. And I want to say also that we think we can go even farther back than diatoms. I told you we found these genes in other organisms. And so the way we think that the CYT genes actually evolved was that, well, I didn't tell you, we found a gene in bacteria that looks kind of like a CYT gene. So we think that this was horizontally transferred into an ancient algal ancestor. And then as the algae diverged into their different groups, they all carried a CYT gene. That's what I defined as the outgroup on that tree. And so we identified it in chrysophytes, silicoflagellates, ciliates, dinoflagellates, and haptophytes. And then when the diatoms diverged, they also still contained that ancient CYT gene, and that is what became their clade B CYT gene. So this is really interesting that to me, that there's this CYT gene that was in the original diatom probably, and it's still maintained throughout the diatom lineages. So it seems to be pretty important. And to look at what the role of this CYT gene is, I'm going to show you how these genes are expressed in some natural field communities. So the data I'm going to show you come from a cruise off of the Washington coast. So here's Washington State and Vancouver Island. And at these 4 locations, we sequenced metatranscriptomes. And a metatranscriptome is very similar to a transcriptome, except that it is all the genes expressed by a mixed community, not just a single organism. So it's a little more complicated to analyze. And what we did is we identified all the CYT gene homologs in that at each location. Then we put those homologs onto the phylogenetic tree in order to identify what type of a CYT it was and what was the most closely related organism expressing that gene. And for you bioinformatics people, the programs I was using were Hummer, pplacer, and then some Python programs. So I wanted to tell you that some of the interesting things about these stations is that at these 3 stations, there was fairly low silicic acid availability. At 4 micromolar or less, the average diatom would be experiencing uptake limitation. Whereas at this location, there was more than twice as much silicic acid available. And the average diatom would not be silicic acid limited at that concentration. So how are they expressing their CYT genes? First, I'm going to show you those 3 locations that had the lower concentrations of silicic acid. And I'm only showing you the genes expressed by the centric diatoms, or at least the sequences that were most closely related to centric diatoms. And on the top are these pie charts that show you the percent of what type of CYT gene was being expressed. So you can see that at these 3 stations, they're mostly expressing CYT-C and then a really small amount of CYT-B. And the numbers underneath represent how many sequences were detected in centric diatoms at that station. So what I'm showing you on the bottom here is the same data just plotted in a slightly different way. I calculated a normalized expression so we could see how the expression levels changed. And here you can also see that they're really just expressing CYT-C a lot more than CYT-B. There's no real pattern between the stations. But now if we look at that 4th station, it completely flips. Now they're expressing CYT-B much more than CYT-C. And that's not caused by any change in CYT-C. CYT-C, they really didn't change how much they were expressing that gene, but they increased the expression of CYT-B. And instead of overwhelming you with tons and tons of data, the multipolar diatoms and the pennate diatoms were doing the exact same thing with their CYT-B genes. They all upregulated this gene at that higher silicic acid station. And what's really cool about having so much data is that we can actually drill down and look at the genus level. So we can say, well, what about the sequences that were most closely related to Cattosaurus? How are those being expressed? And what you can see is that they were doing the same thing at the genus level as we saw at the more broader lineage level too. And I just hope that you guys are excited about this as much as I am, because just a few years ago, this type of thing wouldn't have been possible because we didn't have that much data. But now that the sequencing technologies have increased so rapidly, we have so much data that we can actually start to look at these patterns. I mean, in 2012, I published a paper on sit genes in the environment, and it was based on about 30 sequences. So here we're looking at thousands of sequences, which is really exciting. And so the conclusions, just granted, it's only based on 4 locations, but we have a hypothesis that these clade B-sits are part of this longer-term acclimation to elevated silicic acid concentrations. So it's enabling the cells to utilize a lot of silicic acid when it's readily available. So what we think is that the evolution of sits enabled diatoms to utilize silicic acid resources when they're abundant, and it also enables them to respond to rapid changes in silicic acid availability, which I didn't really show you the data for that, but we came to this conclusion about rapid responses to silicic acid based on some lab experiments that we did. And then the diversity among diatoms may enable them to utilize the same resource in different ways. So having all of this diversification may enable niche partitioning among diatom species. And I just want to bring this back to the carbon cycle where I started. Without these adaptations, diatoms wouldn't be as productive. So the ability to utilize silicic acid in such an efficient way and be able to respond to changing conditions so effectively is what is a really big contributor to them being able to be so productive in the surface ocean. So now we're going to move on to part 2 of the talk and think about the sinking below the surface. So we know there's all this production happening in the surface. There are all these adaptations that enable diatoms and other phytoplankton to be really successful. But what does that matter in terms of carbon export? What is actually getting out of the surface? So to answer that question, we're going to go to the Sargasso Sea. I went out there with my collaborator, Meg Estapa, and also in collaboration with Ken Bissler. And we went out there during 5 different months of the regular time series of the Bermuda Atlantic time series. And when we were out there, we deployed a neutrally buoyant sediment trap. And this sediment trap is designed to excuse me, park at a pre-programmed depth. And we'd put out 3 sediment traps at 3 different depths. And they have these tubes attached to them that are collecting the bulk sinking particle flux. But one of them also has a polyacrylamide gel at the bottom. So this is what that gel looks like. And the nice thing about these gels is compared to just a traditional sediment trap, which has a formalin brine at the bottom, is that when the particles settle into the gel, they stay separated from each other. So you can really identify what they looked like in the same state that they were sinking in. And what I did when we got these samples back is I took thousands of micrographs of them and then ran them through an automated image analysis so we could quantify the number of particles sinking and what their sizes were. So this is just an example of 1 micrograph, how it was processed through that image analysis protocol. So here you can see we can easily count the particles and measure how big they were. So in doing this, we can calculate the particle size distribution. Oh, excuse me, I just need some water. Too much talking. All right, so what you're looking at on the x-axis is the size of the particles, and the y-axis is the flux. And what you can see is that as particles get smaller, they're exponentially more abundant. And the way we defined a small particle here was 11 to 64 microns, and a large particle is anything bigger than that. And I had to work really hard to optically resolve particles that small. A lot of optical methods don't get particles that small, or if they are observed in the water column, oftentimes it's assumed that they're not sinking. So I want to point out that we know that these are sinking because they were captured in the sediment trap. So they had to sink down into that gel. So we wanted to figure out how much carbon do these small particles actually contribute to the total flux. The way we figure that out is I used these measured particle sizes and the measured carbon fluxes to model the carbon content of the particles. And in doing that, we figured out how much carbon is contained in that small particle size fraction. And so along the x-axis here is the percent of the carbon flux by small particles, and the y-axis is the depth that we observed it at. The different colored lines are those different months that we went out there. So what you can see is that these small particles are often responsible for about 20% of the carbon flux out of the surface. So in August, they actually were more than half the carbon flux, and then their influence increases with depth. So the takeaway is really that if we don't resolve these small particles or we overlook them, we're going to be missing a big part of the carbon flux in the ocean and through the water column. And so as a biologist, what I want to know is what are these small particles and how are they getting through the water column? Theoretically, a very small particle should be sinking really slowly. So it's a big question. How are these small particles actually contributing to export? To try to answer these questions, I went on a cruise in the South Atlantic. And so this work was in collaboration with Ben Van Mooy, Ken Bissler, and Sonia Dyerman. So the cruise started in Uruguay and ended in Barbados. And at all of these locations, we deployed a sediment trap, but it was really different than the one that I showed you earlier. This one had a big net on the bottom, and then clamped above it was this frame that had the tubes. And the way we used this was 3 of those tubes were for measuring the bulk POC flux. One had the gel layer, and then this net was used for collecting large amounts of fresh sinking material. And so I want to show you, actually, what that looked like. So we were on the Nore, and this is what that net looked like. And this is what the frame looked like. So in 3 of those tubes, I put a formalin brine at the bottom. So that's just for fixing the bulk POC flux. And then the 4th tube had this gel layer on the bottom. And that enabled us to actually identify the particles.
And the gel's alive, or is it formalin also?
No, it's polyacrylamide.
So it kills on contact?
I am not sure, but I definitely saw zooplankton sometimes like twitching in there, so I'm not sure. Okay, so the net went down to 150 meters, and the tubes were at 125 meters. And then it was tethered at the surface with these floats. And then we just let it float away for about a day. And then we went and found it the next day using the radar. That's what that high flyer at the top is for. So first we got back the tubes, and that was for doing all the quantitative measurements. And then we got back the net, and that was for looking at all of the little particles, but getting a whole lot of them so we could do experiments. And this was the largest particle that we captured. But usually, that only happened once, and it made for a really good dinner. But usually we captured just the particles we were going after. So once we brought them back on, we could filter them down for doing chemical measurements, and then also look at the gel. So hopefully you could see particles in there. And I looked at them under really high magnification so that I could really see what those particles were made of. So what are they made of? Well, what I saw was aggregates, fecal pellets, and then I also saw phytoplankton cells, so diatoms, coccolithophores, and what I'm calling a nanocell, which was just a cell that's about 5 microns. And sometimes I had flagella, but I really couldn't characterize them any more than that. And then a really minor component were the dinoflagellates. And these images are actually of phytoplankton cells that were embedded in the gel. And one thing that I think is kind of amazing is that some of them look actually pretty healthy, like this guy, this Esteromphalus up here, has a lot of chloroplasts that look pretty good. So how did these change across this pretty large region of the ocean? The pie charts represent the change in the particle composition, the numerical composition across the transect. So the dark blue is nanocells, light blue is aggregates, the really light blue is fecal pellets, yellow is diatoms, green is coccolithophores, and gray is dinoflagellates. So you can see that across the stations, there's changes in the composition of the flux. Now, if we compare that to the carbon flux that we measured, there was actually no significant change in carbon flux across this whole region of the ocean, which actually, that's similar to a study from a couple years ago that also measured carbon flux in this region of the ocean. So to me, that's pretty important because the variation in the mechanisms of carbon flux might not be reflected in those bulk measurements. So if you had only measured a bulk POC flux, you might think that the same type of process was responsible for exporting carbon along this pretty massive region of the ocean. But if you actually look at what types of particles are sinking, you can see that there are differences. And that's not going to be resolved by just measuring a bulk measurement. So what did cause the changes in the particle composition? It could just be that the overlying surface phytoplankton community was changing, so that just means there's a different source, and then that corresponds to changes in the flux. But in fact, when we compared them, there was no correspondence between changes in the surface and changes in the flux. So to try to figure out what was driving these changes, I used a principal component analysis. And a PCA, it groups together your observations that explain the most variance. So the measurements I used in this analysis were the surface nutrients, the surface phytoplankton community, and then the sinking particle flux. So it's kind of like looking at your data I had 14, so these are 14 variables. It's kind of like looking at it in 14 dimensions and then projecting it onto a 2-dimensional space that separates your data by the most variance. So what you can see is that these first 2 dimensions explain the most variability, the variance in the data, and it's actually 60%, so that's pretty good. And in this direction of this dimension, it's driven by detrital particle flux and nanocell flux. In this direction are the surface diatoms and silicic acid. This way, and then this second dimension is really much more driven between the difference between diatom flux and coccolithophore flux. So if you look at how our stations grouped, you can see that group 1 is really diatom related, diatoms in the surface and diatom sinking. And you could also think of it also as less influence of detrital particles because it's away from this axis. Group 2 is the opposite. It's really much more related to the detrital particle flux. And group 3 is something more related to coccolithophore flux. So we use this to come up with a hypothesis for the things driving the flux of phytoplankton out of the surface across this transect. And this is just one way to illustrate that. The station 1, or the group 1 locations were driven mostly, they were diatom related. So we think that it was either individual diatom cells that were able to sink out of the surface, or perhaps they were physically mixed down. The other station, or group 2 stations, were much more diverse in terms of the influence of different phytoplankton types, but we think that they were transported by being incorporated into the aggregates or fecal pellets, which perhaps disaggregated after they got out of the surface, and that's how they got into the trap. And then the third group of stations was more related to coccolithophores, but we also think that the aggregation was related to the transport of coccolithophores, although there still could be some role of individual sinking coccolithophores. All right, so now that we've thought about what's sinking out, I just want to briefly talk about some future work where I really want to think about the first part of this talk and the second part of this talk at the same time, and try to put these things together. So one way that I'm thinking about relating surface phytoplankton with their influence on carbon export is by looking at the expression of phytoplankton genes in the surface ocean and compare that with sinking phytoplankton gene expression. And we observed a really awesome diatom bloom at this station here. It was a Coscinodiscus bloom in the Amazon River plume. My collaborator, Harriet Alexander, did a lot of experiments on this surface community to characterize how it responded to nutrient limitations, and she sequenced a lot of metatranscriptomes. What's exciting to me is that this cell was also the main type of particle that was captured in the traps. So we have this really well-characterized surface community that we know is contributing a lot to the flux. And what we did is we isolated that cell, brought it back to the lab, and we did a lot of lab experiments, different growth experiments. And in combination with sequencing the transcriptomes, we also measured how those different growth conditions affected their sinking speed. And this is how we measured sinking speed. We put the cultures at the top of this water column and let the cells sink down. And then when they passed this video camera, we got a video of all these individually settling cells. And from those videos, we can calculate the sinking speed of individual cells. In addition to this, what's also really exciting is we sequenced a metatranscriptome from this sediment trap material. So now we have all of this molecular data from the field and from lab experiments. And we're hoping that this will really help us characterize the molecular factors that indicate things that would induce diatoms to sink. And I recently found out that JGI has agreed to sequence the sediment trap material from all of these locations along the transect. So I'm going to have a pretty huge data set from across this entire region of the ocean, and I'll be able to pair it with the metatranscriptomes from the surface that Harriet has sequenced. So we'll really be able to compare the surface and the sinking phytoplankton. And then the second thing that I'm also really interested in is I really would like to resolve what the relative importance of gravitational settling, aggregation, and physical mixing is in transporting phytoplankton and these other small particles out of the surface ocean. Because we have all these observations showing that these small particles are there, they're getting out, they're a big part of the carbon flux, but it's still not entirely clear exactly how they're getting there. And all of those processes could be really important. So one way that I'm thinking about going about this is, next week I'm going on a cruise with, that'll be a collaboration with a physical oceanographer, Melissa Omond, and a chemical oceanographer, Megan Stappa. And they'll be putting out these instruments that are taking optical and other types of measurements throughout the water column. They profile up and down through the water column, so we'll be characterizing the processes that are going on throughout the water column, and we'll be combining those with the types of particle flux measurements that I showed you today. We're hoping that by starting to do a combined approach like that, we'll be able to start teasing apart some of the mechanisms that are causing these small particles to sink. I'm just showing you this picture because the sediment trap actually has one of Meg's instruments on it. And then and I want to mention that this question is actually a big part of an upcoming NASA project that will be a big field program to look at export. So I'm really hoping I can be part of that too. And kind of apart from that is I'm also really interested in continuing to do some sinking experiments, but I think it would be really interesting to do those with natural communities. and look at how the sinking speed of cells within natural communities changes over time. And this could be done somewhere even like right here in Monterey Bay, where you could monitor how the population is changing its potential to sink, and then compare that with what is actually observed getting out. So does the change in sinking speed actually matter, or do they have to aggregate? And there's been a lot of work in Monterey Bay on sinking particles, so I think there'd be a lot to build on. And then finally, I just want to leave you with this micrograph of my favorite diatom. And I want to acknowledge my collaborators one more time. And these people also were really important in supporting this work. The ships that all the field data was collected on were the Nor, the Thompson, and the Atlantic Explorer. And all those cruises were funded by the NSF. My particle flux studies were part of my postdoc scholarship at Hui. And finally, I just want to acknowledge COSI because they supported me in making those videos that I showed you. Right, thank you.
Well, we have some time for some questions. Go. Jim.
So back to the evolution question. So I can't remember what the silicon history looks like for oceans over millennia.
Yeah, well, yeah, that's something I didn't have time to go into, and it actually is something I've been talking with Ivano about. But there's definitely some papers that hypothesize that silicon levels in the ocean were much, much higher when diatoms evolved, maybe up to 1,000 micromolar. And actually at that high of a concentration, silicic acid just diffuses across the cell membrane. So that's kind of a big question. Why would diatoms even need a silicic acid transporter? When they evolved, they maybe didn't even have to actively take up silicic acid. And I actually think my hypothesis is that that basal gene, the CYP that was in an early diatom, is actually intracellularly localized, so it's actually not in the outer membrane. I think it might be in some of the specialized vesicles that are inside the cell where silica gets concentrated and precipitated in a controlled way. Because even if external concentrations were high, they would still have to control how it's precipitated inside the cell. But that's maybe a wild idea, but I think it makes a lot of sense.
I just wanted you to know how old I really am, because Ginger Armbrust and Ken Bissler took biological oceanography from myself and Jim McCarthy in a former life. And then I was waiting for you to say, and these particles, and hence the diatoms are considered the ballast particles of the ocean, because I really like that word ballast. And then I finally wanted to ask you a question.
Okay.
Live and dead cell or detritus, what's your opinion and quantitative fraction of the two in that bulk number, in that bulk carbon flux number? From looking at the particles as carefully as you did, there was a time when folks thought the bulk of the sinking flux was really the decay products from organism interactions and fecal pellet production and big particles only, but when you've now counted what you call the small things, and you only went to 5 microns at best, is that right?
Yeah. Oh yeah, with the identifications, yeah.
I didn't hear the word bacteria.
No, I couldn't resolve the bacteria.
No, and so I'm dying to know, but you looked at it very carefully.
Yeah.
If you have to, and you made the sizing of it, what fraction of the total carbon would you estimate
Is in the cells? Or
is
living things?
Contributing to the total carbon flux from those diatoms?
Well, I mean, so that is one thing I wish I would have done, but I didn't. And that's how I would do it in the future, is combine the methods I used in the Sargasso Sea with what I did in the South Atlantic. Except what I did in the Sargasso Sea was estimate carbon content based on just size alone. And I think what it would be really cool to do would be to instead of it just being based on size of all particles, but maybe you could separate it out of the size distribution of diatoms, size distribution of aggregates, size distribution, and use those to figure out the contribution of carbon flux from these specific different categories of particles. But how much is alive? I did actually do some growth experiments to figure out if those cells were alive or dead. And at some stations, you could get especially the diatoms to start growing again, and at other stations, not as much. Yeah, but it was the diatoms that would really respond if you incubated that sediment trap material on the deck of the ship for a few days. Sometimes the diatoms would start to grow, so they were alive in there. That's great because I sequenced metatranscriptomes, which is the expressed genes. So that wouldn't be very useful if everything in there was dead, because they need to be alive to be expressing the genes.
Claire.
When you talk about gene expression and you study in the Washington, you look at silicic acid concentrations as controlling, or at least a controlling factor. But what about other factors such as light or growth rate or other micronutrients? You look at any other variables?
Yeah, I mean, it could have been much more than just the silicic acid concentrations. Yeah, I mean, and I don't think that, I mean, using that data was just a way to pull apart, are these genes expressed differently in the field? We definitely don't know for sure that it's caused by the silicic acid concentration difference, and there's only 4 there, so it would be nice if you could do that across a lot more stations. And I think actually I know I have some colleagues who are studying that off of the California coast, actually.
Can one diatom express all of those genes, or is it specific?
So, yeah, some species.
Like one
No.
organism itself, you know what I mean?
No. Some of the pedates have almost all of them, but only the Thalassiocyra diatoms have E. And
Right.
most Thalassiocyra diatoms only have E, but there was one that had a B.
But I guess I'm wondering if like one specific organism can express all, like the ones that it's capable of expressing,
Mm-hmm.
or could it have, you know, A and B in just one cell? Does that make sense? I don't know.
Like, if they have them all, do they express them all?
Like, is it in the genome?
Oh yeah, they are in the genome. Like,
And it's just-
Pseudonychia has 3 different types of CYP genes in its genome. And it expresses all of them, but in different, at different times.
Probably know the answer to the question, but have you done, or the team done an analogous thing with the nitrate transporters, and do you see covariation?
No, but I would love to think about the nitrogen genes too, because they're not just the transporters, but all, like, a lot of the other genes related to nitrogen metabolism are also part of gene families. So I think with this new data set that covers a lot of marine diversity, you could really start to think about it from an evolutionary point of view. And how did those gene families evolve, and how did that evolution maybe enable certain adaptations in different lineages? But no, we haven't. But I would love to.
I was sort of going to ask a similar question, which, since you have access now to the metatranscriptomes from multiple stations, you can look for every gene that's upregulated
Yeah.
in an enriched silicon environment.
Mm-hmm.
And that might be a way to substantiate that the gene expression you're seeing is in fact related to the silica content, because you can put together a pathway and show that, well,
Oh, yeah.
it's a whole pathway, it's
Uh-huh.
not just this one gene, but conceivably could even be other things in a cell.
Yeah. Yeah, I think you could do that. The problem with the silica metabolism is there's not a whole lot known about it. There are some genes known, so I think you could start to get at that, but unfortunately a lot of the silica biology is still totally unknown.
We probably couple it to a lab experiment to show the correlation.
Yeah. So maybe you wouldn't even have to know the function of that gene, but if you saw the same unknown function genes going up at the same time, related.
I have a question. During many of our mesoscale iron enrichment experiments, we've noticed that under iron replete conditions, when cells are growing very quickly, the cell walls of the diatoms are weakly silicified, whereas under iron limited conditions, the cell walls and the tests of the diatoms are heavily silicified. And so some geochemists have jumped on bandwagon to suggest that, under rapidly growing conditions, the diatoms want to be in the surface waters and don't silicify as readily. Yet under limited conditions, they try and ballast themselves so that they'll sink to where there's either more silicate or more iron. What do you say to that?
I thought about that a lot during my PhD, because that study I told you where in 2012 where I looked at sits in the environment was actually out at Station P, and we did a on-deck iron enrichment experiment. And what we found was the way the sit genes changed was that in the iron-limited ones, they were also expressing those B genes a lot more. And so at the time, I thought, maybe when I didn't know as much about the diversity of these sequences across diatoms, I thought maybe that B gene is related to that increase in silicification due to iron. But now, the way I'm thinking about it, it's just a hypothesis, is that maybe the B gene is just related to acclimation to high silica conditions. So if these cells they're growing slowly in the low iron, they're acclimated to this kind of not so great condition, but they still have plenty of silicic acid. So they might be growing slowly, but they have a lot of silicic acid available. So the B gene allows them to just utilize it as much as possible. But then maybe when they're growing fast, when you give them iron and the silica is going to get drawn down, there's not as much to use. So they're not using that B gene, and they're, yeah.
So some combined experiments.
Yeah, I think.
I'd like to wrap it up there and thank Colleen for a great, stimulating seminar, and have at your questions with her after we break. Thank you very much.